Hello, and the topic for this video now, we're going to talk about the Lac Operon. That Lac stands for lactose, all right? If you remember way back now to the beginning of the semester, we talked about lactose being a disaccharide all right, and that lactose can serve as an energy source or a food source for certain bacteria. Oops. An example of bacteria for a name that is going to have the Lac Operon is going to be E. coli, okay? But essentially what lactose is going to be our switch in turning on our genes in the bacteria, okay? So lactose is going to activate transcription. All right? And what I have drawn over here, these boxes, this is going to represent our operon, because remember, I said an operon is a cluster of genes that are physically located together, okay? So this is going to be our operon. If you noticed, I have a little space here. There can be some space between the operon and what's going to regulate that particular operon. So we're going to have a number of different players that we're going to talk about. What we've got here is going to be our repressor, okay? So our repressor is going to be a protein that is always made, and I'm going to draw it just kind of as a mushroomy cap kind of thing here, okay? It's going to have some particular three-dimensional shape. The repressor is always made, all right? And the repressor is going to keep our operon in the off position, okay? Because what our repressor does, because it's a protein, it's a DNA binding protein. Our repressor is going to bind to DNA. And what it's going to do, all right, if RNA polymerase comes along, is it's going to block RNA polymerase from reading the DNA sequence, okay? And our DNA sequence down the road here, there are going to be 3 genes that are important for breaking down lactose and metabolizing lactose. And those are the genes that the bacteria cell is going to want to make when lactose is available. But because bacteria are efficient, they only want to make those products when lactose is present, all right? So the operon is going to be kept off with the repressor, all right? What we would have then is a change, okay? And I should label these quickly so that you know what I'm talking about here. The blue box here, this is going to be our promoter. Our promoter is going to be, just like when we talked before about transcription, that's where RNA polymerase will bind DNA and will start transcription, okay? Our green box here, this is going to be our operator region, okay? The operator region is going to be where our repressor binds or attaches, okay? So that bacteria will keep its operon in the off position until environmental changes environmental conditions change, okay? This is gene 1, gene 2, gene 3. I'm not worried that you know the names of the genes, although I'll talk about them a little bit. If you take microbiology with me, you're going to need to know the names of the genes that break down lactose, okay? But for now, what we've got with our Lac Operon, we're going to have a change, all right? That change in the environment, I'm just going to draw it as a circle. Some nice fluorescent yellow lactose. Suddenly in the bacteria's environment, it has access to lactose. Now, that E. coli wants to switch on the genes that break down lactose so it can use that as an energy source, okay? Our repressor is still made, okay? But what happens with our repressor, I should make this, hold on. Let me do a better shape here, okay? Because we've got our mushroom here. I'm going to make this now a circle. What happens is when lactose binds the repressor, it's going to change the shape of the protein. So now our repressor doesn't fit on the operator region. It can't bind to our operator. So what happens then, our RNA polymerase comes in. And now our RNA polymerase has the green light to start reading that DNA sequence. And what happens is we get transcription of the genes that will break down our lactose, okay? So in this case, when lactose is present, this is going to be our on position, okay? So lactose is the switch to turn things on. When lactose is depleted in the environment, things switch back to the off position, okay? So that's just kind of a basic overview of what happens with the Lac Operon. The genes that are made here, you're going to have a beta-galactosidase that's made. That's going to be the enzyme that's going to be involved in breaking down the lactose. You're going to have also made, associated with lactose, genes that allow lactose to cross the cell wall. And you're going to have genes, this third one over here, we'll call this one the one that's crossing the cell wall. This breaks down the lactose. And you're going to have genes that are going to be important for metabolism, okay? So you've got 3 genes that are made. Typically, those 3 genes, once they're made, will start breaking down lactose. Again, gene regulation, it's all about conserving energy and resources with the cell. You only want to transcribe genes that the cell needs. And that's going to be true for eukaryotic cells also, okay? I know this feels like a lot, but bacterial cells are really much more simple. I'm going to give some generic descriptions, some general descriptions of how genes are regulated in eukaryotic cells in the next video. Won't be quite as much detail, okay? Because it is a lot more complex, all right? If you have any questions, please let me know.